Good morning, everyone. We're so excited to have you here to hear from 4 faculty from the University of Notre Dame. First, Professor Alan Hubner is a data scientist and award-winning statistics educator. He's the Director of Undergraduate Studies for the Department of Applied and Computational Mathematics and Statistics. Second, Professor Mei-Lan Chang leads a team of biomedical researchers to understand the molecular basis of disease and to design small molecules for therapeutic intervention. She's the director of the Chemistry Biochemistry Interface Program, which gives graduate students the opportunity to study multiple disciplines within the fields of science. Next, Professor Laura Fields is a physicist and a member of the Deep Underground Neutrino Experiment, DUNE, that she'll talk about more, where she's a leader in the effort to design, simulate, and characterize the neutrino beam. Lastly, we're going to hear from Professor Jennifer Tank, who is the Gala Professor of Biological Sciences and the Director of the University of Notre Dame's Environmental Change Initiative. Her research was recently featured on the university's What Would You Fight For ad on NBC.
Hello, everybody. Thank you very much. I'm very happy to be here. As Marissa said, my name is Alan Hubner, and I'm in the Department of Applied and Computational Mathematics and Statistics at the University of Notre Dame. Let's just start out by simply asking, what is data? I think that's the first part of understanding what a data scientist does. Well, when you think about it, data is really all around us. It can be numbers, it can be measurements, it can even be audio recordings and video recordings. All those things qualify as data. And if you really want to be philosophical about it, the great statistician John Tukey defined data as a record of experience. So a little more simply, data is created almost in every part of everyday life. Data is created when you buy something. Data is created when you go to the doctor. Data is created when you take an exam in school. All those things are examples of data that is taken on you and your family and your friends pretty much every day of your life. And then in particular, scientists can collect data on many, many things. Just a couple of examples are the climate and weather, animal species, particles and physical experiments, and many, many other things. So getting a little more specific, what does a data scientist do? I don't have a laboratory. I have my office and my desk and my computer and some books and papers and pencils. So I combine math, computer programming, and a couple other fields to get information out of the data. What kind of information do I look for? Well, in biological and medical and pharmaceutical studies, you ask questions like, are new medicines effective? People working at really big companies like Amazon and Facebook are interested in recommending products to customers based on their previous purchases. And people who study social science, they want to understand trends in public health. For example, identify factors that might put individuals at risk for homelessness. So those are just a small sampling of the kinds of questions that data scientists answer using data. So what skills do you need to be a data scientist? Well, first, it's probably not too surprising that you do need to have a lot of math and statistics. It's also probably not too surprising that you have to know how to do some computer programming. And this might be a little more helpful for older students, but in particular, Python is one of the main data science programming languages. And then there's a language called R, just the letter R, and that's really useful, especially if you're doing a lot of statistics. And then you also have to know about databases. You have to have some business skills, and there is a language called SQL that comes in handy when you're getting data from a database. So those are technical skills that I think are not very surprising if you want to be a data scientist. But there's also a lot of other skills you need. For example, you really need the ability to communicate, reading, writing, and speaking. So your English and language arts classes are really important if you want to be a data scientist. And I think some people might be surprised at that. You have to have the skills to communicate to others what the data is saying. It's just not good enough for you to understand it. You have to get others to understand it who might not have the same expertise in statistics and computer programming that you do. You should also be very curious and want to be a lifelong learner. Data scientists are always learning new things because there's always some new technology, some new computer program to learn. And it would also help to have a strong interest in some application area. You need some interest in a field to apply the data science to. For example, it helps to be interested in science or psychology or sports. So some things I do, I apply data science to sports performance science and also to educational psychology. Those are a couple examples. I'll talk about the first one, sports performance science. So I work with the athletics department at Notre Dame, and the athletics department gather all sorts of data on their athletes. And one example are wearable GPS trackers. So this is an example of an athlete wearing a GPS tracker, and it's in the vest. It's the GPS, it's a little piece of electronic equipment, and he's wearing it in his vest there. And those are worn in every practice and every game by almost all the teams, and they take hundreds of measurements every second, every minute, and it gives you just hundreds and thousands of pieces of information about how hard the athlete's working. It tells you about how often they accelerate, how often they decelerate, how often they take a sharp turn. It talks about their heart rate, how often their heart rate is in a certain level. And all those pieces of information are summarized by something called training load, which is a metric telling you how hard the athlete's working. So students and I get that data, and then we use computer programming to what they call clean and process it. In other words, to turn it into a usable form, a process that's also known as data wrangling. And then we apply statistical methods to analyze the data, and then design interactive dashboards, interactive visualizations for the coaches to monitor student athletes, and make real decisions. So for example, this might be a dashboard that a coach would see, and overall, we're looking to prevent injuries. If an athlete is working too hard in practice or in games for too long of a time, they're at risk for injury, and we're aiming to use very clear visualizations for a coach to flag those athletes before they get injured. So this all sounds interesting to you. What can you do right now to prepare yourself to be a data scientist? Well, you should read, especially paper books. Treat everything online as fiction. You want information from paper books. And that kind of goes along with falling in love with this subject, especially science. Be curious. Ask why, like 4-year-olds do. If you're in high school, then you should try some advanced math and computer science courses. See if you like it, but that's not absolutely necessary. And one thing you can do is just try out programming and see if you like it. Python is available for free. You can download it tonight if you have a laptop or a PC at home. And then you can borrow or purchase inexpensively books like Coding for Kids or Python for Teens and Tweens. There's a lot of fun activities out there. And finally, it's a little bit advanced, but if you try that out for a while and you like it, then make a project for yourself. Are you a runner? Then make a program to keep track of your total distance. Do you like to cook? Then make an app that organizes your favorite recipes. So in conclusion, data is all around us, and data science is the topic that makes sense of all of it, and it's a great field, and if you have questions, I'd be happy to answer them. Thank you very much.
Good morning. I'm Meilan Chang. I'm in the Department of Chemistry and Biochemistry, and I'd like to tell you about my research. First, I will like to tell you a bit about my journey. So I was born and raised in Ecuador, South America, and this is the city, some images of the city of Guayaquil on the coast in the Pacific Ocean where I was born. I came to the US to study college, and I went to the University of Southern California in Los Angeles. So I was expected to be a medical doctor. So when I went to USC, they told me there's no such thing as a pre-med, so we'll make you a biology major. So, but then when I took analytical chemistry, that was when it clicked. I thought chemistry was for me, but I then decided to marry biology and chemistry. So for my graduate school, I went to the University of Chicago, where I studied this parasitic plant called Striga asiatica. This is the pretty plant with the red flowers. And this plant parasitizes grasses such as corn, wheat, sorghum. And it forms microscopic seeds that are dormant for years. And in response, it knows when a host is close by, and it germinates and then also forms this organ called a haustorium, in which it attaches to the host to sap all the nutrients. And so my project was to identify the chemical signals that the host put out and figure out how the parasite was recognizing the host. I then went to the Columbia University in New York City. And my project there involved identifications of sugars in natural products. And I used a technique called circular dichroism. I then moved to Dow Chemical in Midland, Michigan, where I studied herbicides and insecticides to see what happens to the herbicides and insecticides in plants, in soil, in the environment. I moved to Upjohn Pharmacia in Kalamazoo, Michigan, which is now Pfizer. And this is one of the drugs I work in. It's called Rescriptor. Generic name is Delabredy. It's an HIV reverse transcriptase inhibitor, and it's on the market. And I look at metabolism of this drug, what happens to this drug in the body of both animals and humans. I then moved to Wayne State University, where I established an office that managed the 200 clinical trials conducted annually at the School of Medicine. And then I came to Notre Dame in 2003, where my research focuses on wound healing among one of the projects, but I also started this training program, which is NIH funded, and is for graduate students to train at the interface of chemistry, biochemistry, and biology. So, moving on to my research, I work on new antibiotic discovery. So this is to treat bacterial infections. And these projects usually start with in silico screening, so computer screening against the bacterial targets. We identify hits, then go synthesize small molecules against that target, then look for activity in vitro assays in the lab. We also look at efficacy, whether that antibiotic works or not, and we have mouse models of infection. And then also we work on mouse and other animals' pharmacokinetics, so what happens to this drug in the body of the animals. And this is an iterative process. Okay, so another project that I work on is to understand why chronic wounds do not heal. So these are diabetic foot ulcers, pressure ulcers, or bed sores. And what we have used is an affinity resin that we synthesize, and this binds to proteins in diseased tissue. And we use both human wounds, so we collected in collaboration with doctors here in the hospitals in South Bend, diabetic foot ulcer, wounds from patients with diabetic foot ulcers and also with pressure ulcers. And then also we established a mouse model of these wounds. And when we identify the proteins in the diseased tissue, we determine the roles of these proteins by using the animal model of disease. And this also includes transgenic knockout animals in which that protein has been depressed. We synthesize selective inhibitors for the identified proteins, and then we use enzyme kinetics, animal models of disease, as well as pharmacology to determine which proteins should be targeted. We have a drug that is scheduled to go into phase 1 clinical trials next year. Okay, so this is another project in my lab, and it's on glioblastoma multiforme. It is a malignant brain tumor. It has very poor prognosis. On average, patients survive 15 months, and the 5-year survival is the lowest of all cancers. It is 5%. This is the disease that President Biden's son passed away from. What we have done is identified an enzyme called MMP14 as the target for intervention. We obtained brain tissues from patients with glioblastoma, and in the controls, and these are the levels of MMP14. We basically do not see MMP14 in control healthy brains, but we see it in the brains of patients with glioblastoma. We have a selective MMP9 inhibitor that shows better survival in a mouse model of glioblastoma and compared to the standard of care. And we are gearing to do a phase 0 clinical trial with this selective MMP14 inhibitor. This is my contact information. You can contact me, or if I can answer any questions, I'll be happy to do so.
So, hi everyone. It's my turn to tell you about my life as a scientist. So the kind of science I do is physics, or specifically neutrino physics. So I'm actually a very new professor at Notre Dame. I've only been there about a year. And before that, I was a scientist at Fermilab, which is what you see on the right of the slide. And I still do research at both places in neutrino physics. So first I want to tell you a little bit about neutrinos. So scientists think that neutrinos are one of the basic particles that make up our universe. And our current model is that there are only 16 particles, or basic building blocks of our universe. And somewhat weirdly, only 3 of those building blocks make up you and me, this auditorium, everything around us. So the up and down quark make up the protons and neutrons that are in the nuclei of the atoms that we're made of. And then electrons orbit those atoms that we're made of. And it's actually a big goal of particle physics right now to understand what all of these other particles, what role they play in the universe. And the part of this that I concentrate on is particles called neutrinos. So they don't make up the matter around us, but there are a huge number of them in the universe because there are lots of things that make neutrinos. So we think that they were made in the Big Bang that created the universe. They're made in supernovas. They're made here on Earth in nuclear reactors, as well as in the nuclear reactor that we call the sun. We can also make them in particle accelerators. And they're produced in any sort of radioactive decay. And it turns out that that happens pretty frequently in bananas. So if you happen to have a banana for lunch, that banana will be emitting quite a few neutrinos. But you don't notice them because neutrinos interact very, very rarely. They can go through light years of lead before they will interact and tell you that they're there. So what this effectively means is that all of these sources in the universe are creating neutrinos and sending them out, and the neutrinos just keep going through all of time without ever interacting, hardly. So hopefully I've convinced you that neutrinos are super weird particles that are deserving of study. So that's what me and my colleagues do. And one of the best ways that we could study neutrinos, so although there are lots of them made in the universe, it turns out that a good way to study them is to make even more of them, which we do at particle accelerators around the world, including some at Fermilab. So I work on an experiment that will send neutrinos from Fermilab in Illinois through the Earth to a large detector in South Dakota. And we can send them through the Earth because they hardly ever interact with anything, so they can go straight through the Earth. And the detectors that we're going to have in South Dakota are really huge. So here's a picture of what we think is going to look like. So the detector is divided into 4 different components. And actually, in addition to being a particle physicist, I'm the mom of 2 boys. So when they were a little bit younger, there was a time in our household where all conversations revolved around dinosaurs, and that's when I made this figure that shows that you can fit 4 T-Rex into one of the 4 modules of this detector. Or if T-Rex is not your preferred distance scale, you can also compare with a great blue whale or a space shuttle to see that this is a really huge detector. And that's actually one of 4 different neutrino experiments that I work on at Fermilab and at CERN, which is in Switzerland, that are all studying neutrinos in different ways. Each of these experiments is run by a collaboration of people. So this is one of my collaborations, Minerva. And when you work in large collaborations, it means that you go to a lot of meetings. So this is a meeting that we had, I think, in Pittsburgh several years ago. Of course, these days in the COVID era, the meetings mean a lot of Zoom. So here's a picture of one of our recent collaboration meetings of the Dune experiment, which is a collaboration of over 1,000 people. So those are always interesting meetings. Other things that I do in my day-to-day life as a scientist, I write a lot of code to analyze data and to help design our experiments. So we use a lot of Python, like one of my colleagues mentioned earlier. I also give a lot of presentations. So this is one thing I didn't realize about being a scientist when I first got into the field, is that you do a lot of public speaking. I probably would have run away screaming had I known that, but I'm actually not too afraid of public speaking these days because I've just had so much practice. I also spend a lot of time advising students who are learning to be young neutrino physicists. So here is a picture from a recent thesis defense of one of my students in Met. And actually getting to see my students succeed and become successful scientists is by far my favorite part of my job. I'll tell you a little bit about how I got to Notre Dame. So I grew up in Arkansas, which is about 10 hours south of here by car. And I am not from a science-oriented family at all. My family, to this day, has no earthly idea what I work on. But I went to college at the University of Arkansas. That's where I first decided to study physics. And then I went to graduate school at Cornell University, which is in upstate New York, the former Vander Fermi Lab at Notre Dame. And I also want to mention, it's not on the slide, but in addition to these places, one of the other cool things about science is that you get to travel a lot. So I've traveled to conferences and to see colleagues in places like Japan and Brazil, Slovakia, England, Switzerland. So it's just a really cool sort of side effect of being a scientist. So if any of you out there are thinking about becoming a scientist, I have a little bit of advice for you. So some of the obvious things that have already been mentioned today is that you should take lots of math and science classes and be sure to ask lots of questions. It's always okay to say, I don't know. I think probably the most important class for being a physicist for me was algebra. A lot of the math that comes after algebra and the physics that comes after algebra is mainly hard because it involves lots of algebra. So you gotta make sure you have that down. Also, if you can, learning to code is very useful. And then also, modern scientists don't really work like you probably imagine Einstein working. We work in these large groups. So working on the soft skills that allow you to interact with other people is important too. So things like when you say you're going to do something, do it. It's surprising how far that will get you in life. Working on your written and verbal communication skills. And then regardless of what you do, it's always good to practice being kind to people that you work with, even if they may be stressed out and not always returning the favor. And then finally, I recommend cultivating mentors. So I definitely couldn't be here today if I hadn't had people who were ready to advocate for me when I was moving through the different steps of my career. So it may not always be comfortable to go stop by your teacher's office or your professor's office and say hi, but it's an important thing to do. So in conclusion, I think being a physicist is not an easy job, but it's something I find fun. And regardless of what you do, I hope that you find it fun as well. And if I can be of any help to you in the future, here are my contact details.
Well, thanks very much. I think I'm the last of our panel. It's an honor to be here in this beautiful new building. My name is Jennifer Tank. I'm an ecologist at Notre Dame. I'm in the biology department. And today I'm going to give you a little bit of a tour of what I do to try and give you an example of how an ecologist sees the world. The field of ecology is about how organisms interact with their environment. So today I'm going to talk about humans and how they interact with fresh water. So first I want to just give us a sense of place. So our sense of place is in the St. Joseph River watershed, and this is an outline of the watershed that crosses from the Michigan to Indiana border, and it means that every drop of water that falls as precipitation ends up in Lake Michigan. And you can see South Bend down in the lower left-hand corner. We're in the southern bend of the St. Joe River, and that's why South Bend is called what it is. But it's a working watershed, and there's a lot of agriculture in the St. Joseph River Basin. And that's something a lot of people don't realize is how hard we're working our land, and that impacts fresh water. So today I'm going to talk to you a little bit about that impact and how we might balance the use of agriculture and the preservation of fresh water. So many of you might have heard about the dead zone in the Gulf of Mexico. It gets a lot of news press in the summers, especially. That's when the dead zone sets up. And it's an area where there's no oxygen in the water. And the fish die and the shellfish die, and that really affects the economy in the Gulf of Mexico. And this has been going on for a while. It's not a new problem. It's an ongoing challenge. And this dead zone can be huge. In 2021, it was 6,300 square miles, which is about 4 million acres of habitat unavailable to fish in the Gulf of Mexico. Imagine how that impacts the fishermen in that area. And this is a really important challenge, but it's a long way from the Corn Belt where we live. But I'm going to show you how they tie together. It's also happening somewhere a little closer to home in western Lake Erie Basin, where we see algal blooms. That's the shot on the right of the Maumee River, which flows into western Lake Erie. That's green, as it's just goopy green during sometimes in the summer. It almost looks like the Chicago River during St. Patrick's Day. And what that green is are algal blooms. And this algae forms because nutrients run off from the landscape and fertilize these algae
eventually the algae, which are little plants, die and decompose, and when they decompose, they take up the oxygen in the water. And so where do those nutrients come from? Well, a big part of it in the Midwest is through fertilizer runoff. We have to fertilize corn and soybeans, which are a lot of the agricultural land use in this region. And corn needs that fertilizer in order to grow, so it's not an option to not use it. But because of that fertilizer impact, when it rains, we get runoff. And so when you see brown mud flowing off of the landscape into waterways, that's often coming with nutrients. And so what we've done is really changed our landscape. This is an outline of the whole Mississippi River Basin. So all that land, like I showed you for the St. Joseph River Basin, all this land drains, that outline drains into the Gulf of Mexico. It's a huge area, and a big hot spot of that area in yellow is agriculture. And that's where this fertilizer runoff can really impact what's happening in the Gulf. So the challenge is we've changed the vegetation to agriculture. We've lost a lot of our natural filters like our wetlands and forests. And in order to sustain productive agriculture and minimize nutrient runoff, we've got to think of some different ways to do business. And that's a little bit of what my lab does. And so how can agricultural conservation, which are practices that farmers do a little bit differently than normal, minimize nutrient runoff while still maintaining their profits? So two practices we work on is to document how year-round vegetation, as well as floodplains as filters, help to minimize that runoff. So I'm going to talk first about the planting of cover crops and then the restoration of floodplains. So first on the fields, farmers can do something a little different. Instead of bare ground in October after they harvest the corn or soybeans, they can plant something else. And that makes like a blanket on the land that keeps that runoff from happening during storms. Basically, the little saying I use is keep green on the ground year round. And it's actually the root systems of the green vegetation that helps keep those nutrients on the fields instead of in the water. And we've done some big experiments, used the landscape as our laboratory. Part of the Indiana Watershed Initiative, which I run, has incentivized the planting of cover crops. We planted 672 football fields worth in a watershed in Indiana just to see how it would impact water quality. And basically the take home is cover crops reduce nutrient runoff. So my students, young ecologists, what they do is sample the water that's coming off of the fields and in the waterways year round. And you can see a picture of my student Amelia, and it was right around January, a couple of years ago. This is what her sampling site looked like, deep in the snow. We've got fall sampling. They sample every 2 weeks year round. And what we found is it really reduces nitrogen loss to waterways. With cover crops, the green bar you can see is a lot less than the brown bar when there's bare soil. But then sometimes nutrients do get into the waterway. Is there anything we can do to waterways to make them more like sponges instead of like pipes? And another way is to restore floodplains. So the top left shows a conventional ditch, which is just like a big pipe that moves water quickly downstream. On the bottom left, you'll see the two-stage ditch, which has mini floodplains. And on the right-hand side, you can see where we actually built that on the landscape to see how it would impact water quality. And here's what that construction looked like. And you can see that when we're building it, well, I didn't build it, but we paid someone to build it. It cost $10 a foot, and we built 4.1 miles of it. So we really did a whole watershed experiment on this. And you can see down in the lower right-hand corner, the waterway doesn't look very different once it recovers, but those floodplains underneath do a lot of work. They work during storms. So here's what that waterway looks like when that's filled with water during a storm. But look at the difference in the water at the bottom. You can see it in that clear jar compared to the upstream, which is that cloudy jar. And that removal of those particles, the floodplains do that work, and that really improves water quality downstream. And then the biology is working as well. That wider ditch has a lot more biology, and it removes a lot more nitrogen because of the microbes that take up that nitrogen. So the goal is to really be able to achieve this type of watershed scale impact on the landscape. But really the key are young ecologists. And so that's what I spend a lot of time doing. As an ecologist, I train other ecologists. So I'm training the next generation of environmental stewards. And these are some of my students in one of my courses sampling over at Juday Creek near St. Pat's Park. In order to scale up what we're learning in our lab at Notre Dame, we really need to send these ecologists out into the world and have them try it out in the regions that they're in. And so that's what we've been doing. Our goal is to take that dead zone that you see in red at the top of the screen and shrink it down. We want to shrink it down to what the government is asking us to do, or even further, down to no dead zone. And that's going to take a lot of work, a lot of people out doing this kind of innovative freshwater, and I call it sowing the seeds of change. So this is a group of my former students who are all over the country now working on how land use impacts freshwater, and hopefully we'll see a change. So if I could just end with what I would advise students, and pretty much not just for ecology, but anyone who wants to be a scientist, is to always know in your heart that your science matters. When you work with others, be a generous partner. As you've heard from the other scientists on the panel, we don't do this job alone. It usually involves collaboration, and being a good partner is a big part of that. The third thing is determination and grit. So stay the course when things get hard. You'll always get to the finish line. You just have to keep moving towards it. And finally, it's hard work to be a scientist, but if you love what you do, as they say, you never work a day in your life. So thanks very much.